versus codec. So that is the most commonest lesion that is going to affect the younger age group. Because of that, they are more known for developing this colorectal carcinoma. Okay, males are more known for developing this colorectal carcinoma. Okay, so most commonest etiology is dietary, mostly this low fiber diet, high carbohydrate diet, and high fat diet. Okay, this is most important. Pre-existing lesions like polyps and this inflammatory bowel diseases like Crohn's disease and ulcerative colitis. Those patients are more prone for developing colorectal carcinoma. That is the most commonest and most important etiology for development of this colorectal carcinoma. Okay. So other than that, bacterial, mostly this clostridial organism, because these organisms are going to produce some nuclear deaminase enzyme that's going to cause some mutation and can develop into malignancy. Okay. Other than the surgery, like cholecystectomy, gastric surgery, the cost for this is still unknown. Okay. Next one, irradiation during the childhood. These two symptoms are commonly associated with colorectal carcinoma. One is H syndrome. Here, there will be hereditary non-polyposis colorectal cancer syndrome. Most commonly, it is going to happen in the younger age group, that is around 20 to 30 years. Okay. Other than that, Thorne syndrome. Here, patient will be having colorectal carcinoma. Along with that, they will be having multiple sebaceous carcinoma and keratoacanthoma. Okay, these are the two syndromes that are associated with developing of colorectal carcinoma. Etiology, anyone is getting it out? Etiology. First common is diet. Second thing is your pre-existing conditions like Crohn's disease, ulcerative and colon. Next thing is your clostridial bacteria. Next one is your surgery. Last thing, these two symptoms are associated with developing colorectal carcinoma. This is Thorne syndrome and Lynch syndrome. Okay. Etiology is different. Pathogenesis is different. Etiology means because of this, these factors, malignancy can develop. Pathogenesis means these are the mutations or mechanism are happening to develop into a cancer. Okay. So this is the most commonest genes involved. Okay. First common gene is your APC gene. What is APC gene? Adenomatous polyposis gene. Okay. Next thing is your common gene, that is P53 gene mutation. There will be loss of P53 mutation. P53, what is the function of the P53 gene? So, in the cell cycle, these P53 acts as a checkpoints. Okay, there is any abnormality or abnormal mutation in the cell is being identified. This P53 is going to repair the affected cell, or it is going to. What is the other mechanism of P53? Either cell can be repaired or induce apoptosis. Okay, that is the function of P53. Here, that thing is going to be, that function is going to get lost because of the excessive proliferation of cells you are going to see. Other than that, KRAS gene mutation. Okay, and telomerase activation. What is the function of telomerase?
Cellular aging.
Cellular aging. So this telomerase is going to cause cellular aging. So telomere, this activation is going to decrease the cellular aging. Because of that, cell is going to get proliferated more. So ultimately, all these things are going to cause increased cell proliferation and human development. Okay. Another commonest important pathogenesis for this colorectal carcinoma is your defects in the DNA mismatch repair gene. Okay, that is also a commonest mutation is going to happen. Okay, defects in the DNA repair genes. So DNA repair genes, what is the function? In the name itself, it's there. It's going to repair the cell. Okay, simple. If that gene is going to get affected, what is going to happen? The repair is not going to happen. Because of that, cell proliferation is going to happen. Okay, ultimately for all these things, the end result will be increased cell proliferation and tumor formation. Okay, any doubts in this? These three are important. One is APC, RAS and P53. Other than that, telomerase activation. These four things are most important. Any doubts in this? So two pathways. One is suffix engine mismatch suffix pathway. That is going to be a second next slide. Here, this is the commonest thing. Is going to commonest mutation that is going to happen in the colorectal cancer. Okay. This is a normal colon. Normal colon contains 4 layers: mucosa, submucosa, muscular layer, and outmost layer is the serous cell layer. Okay. First thing, there will be APC gene mutation will be there. Okay. Because of that, this mucosa is going to get exposed, and that mucosa is going to be at risk of developing malignancy. Okay. Next mutation, there will be APC mutation. Along with that, beta catenin gene mutation is going to happen. Okay. Because of that, there will be development of this adenoma. Okay. Once adenoma has been developed, additional mutations will be there. That is KRAS gene mutation, P53 gene mutation. Okay. That is the third mutation is going to happen. Finally, after developing into adenoma, this adenoma is going to develop into adenocarcinoma. Adenoma is just a epithelial proliferation. Carcinoma, here cell proliferation. Along with that, the cells are going to what has happened here? Invasion has happened. Okay. From mucosa, the cell is going to get proliferated and it's going to invade the uppermost layer, that is muscular layer, serosa. Along with it, it's going to involve the other sites. Okay. For that, telomerase gene activation will be there. Okay. So initial mutation, APC gene mutation. Because of that, mucosa is at risk of developing into malignancy. Next thing, APC and beta catenin gene mutation will be there. Okay. Because of that, there is development of adenoma. Once adenoma is developed, additional mutations are going to happen. That is P53 gene mutation, KRAS mutation. Okay. Both of the genes are being suppressed. So, increased cell proliferation is going to happen. So, finally, telomerase activation is going to happen. The tumor cells are going to proliferate more and it is going to involve the deeper layer. Okay. Whatever I told, that is only is more than enough. This is not you need. This is going to be 12 otherwise that not you need. First will be APC mutation. Second will be beta catenin mutation. Third will be KRAS and P53 mutation. Last will be your telomerase activation. Okay. Any doubts in this? So if you are having any doubts, you can ask in your textbook. Will be very complicated.